Good morning or good afternoon, depending on where you are in the world. My name's Alex Jones, and I'm the Chief Commercial Officer for Nanolive, with the honour to welcome you to this special event. Today, Nanolive is excited to present the live T Cell Assay, a unique multi-parametric live cell assay that allows researchers to interrogate the entire process of T cell killing and enables the selection of the best T cell therapies using patient-derived living cells in a completely non-invasive and label-free fashion. But rather than hear that from me, I'm joined by some of my colleagues who've made this possible and by one of our industry clients. First, allow me to introduce Dr. Mathieu Frechin, Nanolive's head of AI quantitative biology. Matt and his team developed the science behind this amazing tool. Hi, Matt.
Hi, Alex. Thank you very much for the kind introduction. Looking forward.
Thank you, Matt. Following Matt's presentation, we'll be joined by scientific communications specialist, Dr. Emma Gibbin-Lameira, who you might already know from our webinars. Emma will share some real user cases where we tested immunotherapy candidates in our lab in collaboration with some biopharma researchers. Hello, Emma.
Hi, Alex. Thanks. Excited to be here.
Thanks, Emma. After Emma, it'll be the moment of Kulsum Farshori, one of our extremely talented technical sales specialists. Kulsum will give you a short demo of the tool, showing you how to get these unique metrics at the click of a mouse. Kulsum will go through the full process of segmentation and analysis, showing you the richness of our metrics. Hi, Kulsum.
Hi, Alex. I'm happy to be here.
Thanks, Kulsum. Last but not least, you will hear from Valery Moine from Lightchain Bioscience, or LCB. LCB is a Geneva-based company developing bispecific antibodies that rely on their light chain to function. Valery will describe how this technology can help select their best drug candidates earlier and faster. But before handing over to our presenters, I'd like to show you the world premiere of our product video. Ladies and gentlemen, the live T Cell Assay.
Nanolive presents the live T Cell Assay for multi-parametric, non-invasive immunotherapy analysis to select the best T cell therapies on living cells in a completely label-free fashion. The live T cell assay allows you to follow dynamic phenotypic changes in target cell populations and T cell populations over long periods of time. In addition to providing novel information about target and T cell interactions at every time point. Nanolive's imaging platform, together with the live T cell assay, extracts continuous, multiplexed, and unbiased data to reveal how T cells find, bind, stress, and kill their targets. Use T cell speed and distance to target to measure how your experimental treatments impact the duration of the finding phase. Compare how drug treatments perform against their targets in vitro by calculating the duration of the binding phase. Evaluate when your T cells start affecting your target cells by monitoring changes in the internal composition of your target cells through granularity. Identify the precise moment your T cells start to kill their targets, whether it be within minutes or hours. Finally, once your cells have killed, are they able to kill again and again? Another advantage is the production of a comprehensive report which summarizes key temporal events and statistics describing the T cell response in a simple, user-friendly manner. Live T Cell Assay from Nanolive, the only solution to provide a complete, quantitative analysis of T cell dynamics from start to finish. Select your best candidates earlier and faster.
Wow, I hope you enjoyed that. Now we're ready to start. Please note you can ask for a quote at any time by typing quote in the chat box, and you can also ask questions, of course, by typing on the question box. Nanolive's policy is to moderate discussions during live meetings, so your questions will be sent to us by email and we'll reply to you once the event will be over. So, Matt, are you ready?
Thank you very much, Alex, for your introduction. A warm welcome to all the attendees. I am Dr. Mathieu Frechin, Head of AI for Quantitative Biology, and I am very happy to introduce you to our new product called the Live T Cell Assay. My aim for today is to present the context and challenges that led to the creation of our new product, which allows for the first time to analyze the interaction between two different cell populations captured using holotomographic microscopy, such as the interaction displayed in this movie between T cells and cancer cells. The cells of those two different populations can be segmented separately, completely label-free, single T cells on one side, shown in color here, and single target cells on the other side, shown in gray. Those segmentation can then be used to produce biologically relevant analysis, over which we will come back later. For those who are not experts, T cells are fundamental for an efficient immune response. They are produced in the bone marrow, mature in the thymus, and then are released into the blood, lymph nodes, and lymph. The main role of T cells is to kill anything that does not belong to the body, such as cancer cells, for example. In this image taken using an electron microscope, you can see T cells, such as this gray cell, creating immune synapses with a cancer cell in yellow. The image is stunning, but unfortunately it is only a snapshot of a broad and complex dynamic process. T cells have many roles besides killing cells. These can be split in two broad functions. First, killing non-self through direct cell-cell contact, as we've just seen, but also triggering complex downstream immune responses through cytokine secretion, suppressing immune responses, which is key to avoid autoimmune reactions, and memorizing non-self material to trigger quicker immune responses. So we kept this in mind to create our new product. The Live T Cell Assay characterizes how T cells generally interact with their target cells. But of course, it also has the capacity to quantify T cell mediated cell killing. Let's go back to the movie I briefly showed previously. This is a label-free movie produced in-house with our collaborators. The imaging tool used for the acquisition is Nanolive's automated microscopes, the 𝐏𝐫𝐸. The video shows freshly extracted human T cells mixed with colorectal cancer cells. These cells have been treated with B-specific antibodies to promote and stabilize the contact between the two cell populations, the T cells and the target cells. The action of those T cells were observed for 25 hours at a frequency of 1 image every 2 minutes over a field of view of 450 times 450 micrometers. The movie is obviously packed with information, and our new tool is designed to extract them in a completely automatic fashion, providing first a sharp segmentation under the form of masks of T cells and target cells. Secondly, multiple single-cell metrics for each cells within those two populations. And third is specific metrics for cell-cell interactions, which are specifically designed to describe how T cells and target cells play together. Moreover, to help evaluating and to navigate through these metrics, Nanolive proposes an additional advanced report containing the timing of key events such as binding of T cells on target cells, killing or target cells anti-proliferative action. We also provide novel metrics such as the speed of cell death or the number of target cells a T cell can kill. This allows to characterize an elusive T cell property known as the T cell serial killing capability. So this is only a very brief example of some of the metrics we can provide, and Dr. Gibbin-Lameira will go into more details in the next presentation. In the remaining time, I will cover the computer vision approach used to enable the analysis of T cell and target cell interactions, and why this tool is actually a must-have and a key addition to the solutions that are currently available for immuno-oncology. Nanolive recently released a fundamental cell segmentation and analysis tool called 𝐬𝐩𝐞𝐚𝐦. This tool provides general metrics such as cell size, shape, dry mass, or texture measurements. It is an amazing tool to analyze cell monocultures, but it is not really designed for a mixed culture of two cell lines, such as effector T cells and target cells playing together. We therefore worked on a new strategy to rapidly segment effector T cells on one side and target cell on the other, both being in the same time-lapse experiment. To do this, we had to find a specific and complex object signature that goes beyond direct measurements such as shape descriptors or size or intensities or even texture. We succeeded, and we can proudly say that today we can discriminate with precision effector T cells from their target cells, even when small dead cells appear throughout a time-lapse experiment. Computer vision enhanced by today's latest machine learning techniques can be very powerful when combined with Nanolive's holotomographic microscopy technique. In this video, you can observe naive human T cells interacting with macrophages for approximately 30 hours. In fact, macrophages are potent antigen-presenting cells. T cells can also trigger macrophage death when one is found to be too old, and this in order to keep homeostasis within the human body. This is what happens in the center of this field of view. You can see the precision with which T cells are detected, even if there is an overlap between a target and a T cell. Our AI-driven detection allows for robust segmentation with no need for module tuning or long setup time. The user simply needs to mix their T cells with their target cells and to immediately start time-lapse imaging. At the end of the experiment, a single click is enough to launch the analysis, which segments the effector cells and the target cells and calculate both the interaction metrics and the advanced reporting from the large dataset we produce. Again, Dr. Gibbin-Lameira will give concrete examples, and later Kulsum Farshori will execute a demo of the software. So, by looking at both cell types at the same time, without any labels, and by quantifying cell-cell interactions, our solution offers a new angle for looking at the relationship between an effector T cell and its target cell. One that is truly non-invasive for the T cells, and that will leave the sample ready for further analysis or usage. This software is designed to enrich and complement the information provided by state-of-the-art tools that currently exist for in vitro immuno-oncology drug candidate characterization, functional assays that are used to detect immune checkpoints, quantify cytokine, measure the affinity or avidity of effector T cells for their targets, or simply for measuring target cell death, are all designed to analyze a precise quality of the T cell and target cell relationship. Our solution brings a broader view of the full process, expanding the set of solutions for biopharma and researchers. I will now briefly conclude before giving back the microphone to Alex and for the next presentations. The live T Cell Assay demonstrates the power of transdisciplinary innovative effort. By using advanced AI and computer vision with the extremely rich images that are produced by Nanolive's microscopes, our live T Cell Assay will offer new solutions to the field of immuno-oncology and address open therapeutic and research challenges. So back to you, Alex.
Very impressive. But how can this tool be used in real user cases? So Emma, can you tell us more?
I sure can, Alex. So firstly, thank you for joining us, and thank you to Nanolive for giving me the opportunity to talk to you all today. Matt did a great job in explaining the challenges his team faced and the innovative solutions they developed. Now my job is to contextualize and explain those results. To do this, my talk will be split into 3 parts. I will begin by using a case study to explain how temporal information in the report, such as binding and killing, are generated. Then I'll present 2 use case studies where I use the live T cell assay to quantify the effect of drug addition relative to a control, and then to compare the performance of 2 candidate drugs. I will finish with a brief discussion and then some conclusions. So let's get started. For the first part of my talk, I will use the dataset that Matt introduced you to earlier. So just to remind you, this dataset shows how a bispecific antibody influences T cell and target cell interactions over time. The video on the right shows the refractive index images which were collected every 2 minutes for 25 hours using Nanolive's automated microscope, the CXA. Running this dataset through the live T cell assay produces the following report. Binding was detected after 34 minutes and killing after 3 hours and 14 minutes. These are followed by a range of descriptive metrics summarizing the data. But how are these temporal events detected, and what do the additional metrics mean? We've prepared a video to guide you through the full timeline of the T cell response, from start to finish. But before I start the video, I want to take a moment to orientate you to the slide. So on the left of your screen, you can see the same video as you saw before, but with the segmentation masks that have been generated by the live T cell assay overlaid on top of the refractive index images. Target cells are coloured blue, T cells in red, and the positions where they overlap or interact are coloured in yellow. On the bottom left of the screen, a timeline will guide you through the different stages of the T cell response. And on the right, metrics relevant to each stage will be shown. I will loop the video each time we see a new metric so that we can walk through the T cell response together. So the first stage is the finding stage. This stage lasts from time 0 until binding is detected. There are 3 metrics that are important for defining the find-to-bind transition. These are T cell speed, the ratio of T cells that are in contact with their target cells, and the average minimum distance between T cells and target cells. So during this phase, speed and distance to target decline, while contact between T cells and their target cells increases. And at the moment that these 3 metrics stabilize, we enter the binding phase. Now, binding is critical to T cell function, and optimizing the binding properties of T cells lies at the heart of developing immunotherapy strategies such as chimeric antigen receptor or CAR T cells and T cell receptor modified T cells as well. Identifying the timing and duration of the binding phase is an important indicator of the performance of engineered cells. So in this case study, binding lasts for 2 hours and 36 minutes before the first kill is detected. The next step in our timeline is the stress phase. We use a metric called granularity to detect phenotypic stress in target cells. This is a texture metric that describes the internal composition and structure of cells. Intact cells have a higher granularity because their organelles are well-defined and organized. When cells become stressed, the organelles become fuzzy and disordered, and so granularity decreases. Stress can occur during the binding or the killing phases. In this dataset, it coincided with the killing phase. So here we see the first cell that's killed in the dataset. This occurs at 3 hours and 14 minutes. There are 3 metrics that are important for identifying the start of the killing phase. These are mean target cell area, mean target cell dry mass, and target cell confluency. Area and confluency obviously decrease during this phase, while dry mass density increases as the cells shrivel when they die. Other important metrics contained in the report are derived during this phase. T cell anti-proliferative action is calculated from the moment target cell confluency starts to decline, while analysing the rate of change in all 3 metrics allows us to estimate when the fastest killing rate is achieved and when T cells enter the exhaustion phase. At the end of the experiment, we can calculate the total number of target cells that have been killed and 2 other parameters, which we will see later in the use case study. So now we're all experts on how the data is calculated and what the metrics mean. Let's see how we can apply this. In the first use case, I will show you how we can quantify the effect of drug addition relative to a control. In this example, human cancer cells and patient T cells were exposed to either a different bispecific antibody or a control antibody. Images were collected every 9.25 minutes for 6 hours using the 5x5 grid scan mode on a CXA, and 3 experimental replicates were conducted. The resulting datasets were run through the live T Cell Assay and analysed as the mean relative change between the experimental treatment and the control at each and every time point. So in these graphs, the x=0 line represents the performance of the control antibody. When I play the video, you will see that the antibody initiates a fast and strong T cell killing response. In contrast, no killing is detected in the control. The decline in normalized target cell confluency relative to the control, which reaches 22% at its greatest different point between the two, confirms that the drug is having a positive effect on the T cell response. Examining the interactive metrics sheds some light on how this antibody functions. The ratio of T cells in contact with target cells was on average 32% higher when the bispecific antibody was present, while the reduction in both T cell velocity and the average minimum distance between T cells and their targets was reduced. These results lead us to suggest that antibody addition has a positive effect on T cell killing capacity, possibly by improving the efficiency of T cell binding by bringing T cells and their target cells closer together. In the second use case, we will compare the performance of 2 antibody candidate drugs, which we will refer to in this example as antibody 1 and 2. Killing was detected in both antibody treatments. The timing and rate of cell killing, however, differed substantially between the 2 antibodies, with antibody 1 killing cells much faster and at a much greater scale than antibody 2. Again, no killing was detected in the control, and so this was subtracted from the performance of the antibody, so everything is relative to the control. The results of the quantitative analysis will be shown as before, except that this time the response to antibody 1 will be provided in green on the graphs, and the response to antibody 2 will be coloured in orange. So if we start the movie, in the first graph we can see that antibody 1 increases the ratio of T cells in contact with target cells, again relative to the control. The second graph shows that it also reduces the distance between target cells and their T cells, again relative to the control. And the high killing efficiency of antibody 1 is nicely captured using the following two metrics, which are absolute metrics, not relative to the control because we don't see any killing. And these two metrics are the mean number of dead target cells per hour and the number of target cells killed by a single T cell every hour during the experiment. If we now add the performance of antibody 2 on top, we can see that there's a similar increase in the rate and magnitude of the ratio of T cells in contact with target cells within the first 6 hours of the experiment. But this value declines over the rest of the course of the experiment. The second graph perhaps explains why. We can see that antibody 2 is not as efficient at reducing the distance between T cells and their target cells, i.e. it's higher than the line in antibody 1, even if it's still below the control. In fact, if we compare the average minimum distance over the first 6 hours of the experiment, we can even put a number on this difference. So antibody 1 brings T cells and target cells 1.31 micrometers closer on average than antibody 2. Considered together, these two metrics could explain why the mean number of dead target cells per hour is so much lower when antibody 2 is used. As the graph appears, I would like to take this opportunity to explain a little bit further about what we see in this graph. The negative values at the start of the experiment represent the time lag between the addition of the antibody and the response of the T cell. When this value becomes positive, T cell activity becomes anti-proliferative. This occurs at around 3 hours with antibody 1 and around 6 hours with antibody 2. The difference in the serial killing ability of T cells is also pronounced between the two. It's not detected until after 9 hours with antibody 2, but occurs as soon as T cell activity becomes anti-proliferative when antibody 1 is used. So these results would lead us to conclude that antibody 1 outperforms antibody 2 by reducing the distance between T cells and target cells and by increasing individual T cell serial killing capacity. In this example then, we would thus proceed with selecting antibody 1 as the best candidate for development. The three examples I've shown you today are just a tiny fraction of the possibilities the live T cell assay has to offer. However, what they do show is that we're able to generate comprehensive profiles of T cell and target cell interactions, and we're able to compare how drugs change these profiles relative to either a control or other drug candidates. How you use the assay really depends on your needs. If you want a quick, comprehensive report of T cell and target cell interactions that's based on label-free and artifact-free data, then the automated report is available. But if you want to dive into the details of the response and the mechanism of action of your drug, then you can do that. The metrics are there and available, waiting for you to explore. The choice is ultimately up to you. So unfortunately, I have to leave you there, but I would like to thank you very much for your attention, and I hope you enjoy the rest of the program. Back to you, Alex.
Incredible results indeed. We'll hear later from one of our biopharma users and what he thinks about that. But now let's take a closer look at the software itself. Kulsum, I believe you're ready to give us a virtual tour of the user experience. But what does it take to realize such an insightful analysis?
Yes, I'm happy today to introduce you to this software and how to get easily from the acquisition to this analysis at the click of a button. So to start off with, we have the 𝐸𝐸 software dashboard over here with the different options, first of all, for acquisition with the CXA microscope, then the options for analysis, and at the bottom, the application modules where we have the different protocols to analyze data. Today, we will focus on the T cell killing assay. First of all, I will open a data set that was acquired with Nanolive CXA microscope. This data set has some target cells and T cells in co-culture, and this acquisition was done over 25 hours with an acquisition frequency of every 2 minutes. You will appreciate here that this is all label-free data, and you can see the interactions between the T cells and the target cells over this duration. As I go through the time points, you will see an initial proliferation of the target cells, but then there will be more interaction with the T cells, followed by some killing. You can see there are many, many time points and a lot of data that has been acquired with this microscope. It's extremely rich, and that's the beauty of the CXA. At the end, if I scroll through till the last time points, you will notice that most of the target cells are dead. And there are still some T cells up in here floating around. We will now analyze this data. To do so, you just have to click on Analyze. Then choose the protocol that you would like to use, in this case, the T Cell Assay. And just give a name to the dataset. Once you start your analysis, the software does everything else for you. It will compile the images and then move on to the segmentation. And there's nothing else that you have to do as a user. So extremely easy, user-friendly, single click. And we will now appreciate, as I open this data set that has been done, since this can take some time depending on the computer power that you have, you will notice that the segmentation masks are extremely precise. And you can then isolate not only the single T cells, but also the target cells. Considering that these are completely label-free, this is very, very precise. So here I have the mask of the T cells. You can see each of these individual specks is a single T cell. And if I move on to the mask of the target cells, you can see that there. So once again, this is all label-free data, and we can segment the T cells and the target cells all individually. And thanks to this really precise segmentation, we can also get further information, such as here you notice in blue, you have all of the target cells, and in red and yellow, you have the T cells. What are these colors? Basically, whenever a T cell is not in contact with the target cell, it is red. And when you have the contact with the target cells, such as here and here, it will be shown in yellow. And this way you can get a very precise overview of what is going on during the experiment and how many T cells are interacting with the target cells. If we go all the way to the end of this experiment, when the cells are dying, you can really appreciate that even though the target cells are dead and can look morphologically similar to T cells, the software still manages to segment them and differentiate them. And that is due to Nanolive's proprietary algorithm. So it is an extremely precise way to segment completely label-free data without the use of any other markers. Now, with all this precise segmentation, we can now get nice analytical data out of it. So if we move on to the Analysis tab up here, you can see the different metrics which are available. So first of all, you have all of the basic metrics from the standard analysis protocol, which show for both cell types, T cells and target cells, for example, the cell count, or then the compactness, the granularity, etc. But then for this special live T cell assay, we can also get specific metrics which are specific to this type of assay, a killing assay, such as the number of T cells in contact with the target cells. Here we can appreciate that in the beginning of the experiment, they were quite low, of course, since they are not yet interacting, and the interaction increases very, very quickly and continues to increase all the way till the end of the experiment. We can also get other metrics, such as the target cell area, which is covered by T cells, which is also a nice way to understand which percentage of the area is covered and how much interaction is going on. So this is just a basic overview of what can be done with this software. You can see it's extremely easy. All of the results can be exported as well, all of the masks and all of the metrics in CSV format at the click of a button. This can also, of course, be done in the 96 well plate format, because of course you need to get many more conditions to get statistically relevant information. So here I have an example of an assay done in 96 well plate format. And again, here you can appreciate how the T cells have been very nicely segmented from the target cells. And once again, the overlay of the two. And this can be done in many wells in parallel. So that's all there is to it. It's an extremely easy, user-friendly software, and it gives you very valuable metrics at the end. I hope you enjoyed this short presentation.
Thanks, Kulsum. Very straightforward. So now finally with us Valery Moine, Head of Pharmacology Unit at Lightchain Bioscience, and he will relate to us how this technology can accelerate drug development. Valery is unfortunately not able to be with us here live, so this interview has been pre-recorded. Hello, Valery. Thank you very much for being with us here today. Firstly, could you briefly tell us a little bit about Lightchain Bioscience, what your company's mission is? I know most of your bispecific antibodies are focusing on cancer targets. Can you tell us a little bit about how that works?
Yeah, hi Alex. Lightchain Bioscience is a brand of non-immune assay, and we are located in Planowet, close to Geneva. Non-immune has brought 7 monoclonal antibodies to clinical development stage, one of which, 𝐬𝐸𝐞𝐚𝐬, has been approved by the FDA in 2018. After the successful divestment of 𝐬𝐸𝐸-related activities, we now focus on our bispecific technology to build a pipeline of multi-specific antibodies under the brand Lightchain Bioscience. Most of the programs in our current pipeline are dedicated to fight cancer. At Lightchain Bioscience, we explore 2 different strategies to treat cancer. The first strategy is to selectively block the don't-eat-me signal on cancer cells and to mobilize the tumor-associated macrophages and natural killer cells to kill cancer cells. Cancer cells, CD47, conveys a don't-eat-me signal to better escape the immune system. A large body of evidence indicates that blocking the activity of CD47 with monoclonal antibodies restores killing of tumor cells. One major difficulty is that CD47 is expressed on every cell of the human body. To overcome this limitation, we have developed a unique bispecific antibody approach to selectively block CD47 on cancer cells, leaving normal cells unaffected. The second strategy is to redirect T cells to kill cancer cells. In this case, one arm of the bispecifics binds to the T cells and the other arm to tumor cells. By ligating T cells to cancer cells, the bispecific can stimulate the T cell to kill tumor cells.
Thanks. So may I ask, you're one of the first people to actually see and use the live T cell assay from Nanolive. So how does that compare with assays you've maybe used in the past, and what extra benefits does it bring you?
Firstly, I have to say that it was the first time that I was able to observe so nicely the killing process of the cancer cell by T cells. The pictures, videos are very resolutive. I can observe the cell-cell interaction between T cells and cancer cells and the modification of the cancer cell shape during the killing process. In our lab, before working with Nanolive, we use an indirect method to quantify the cancer cell killing, which does not show the killing process. By using the Nanolive technology, we can nicely illustrate the mode of action of our bispecifics by showing the live imaging videos. The advantage of this technology is that we don't need to stain the cells involved in the assay, and it's important because the staining step could affect the functionality of the effector cells. In addition, to show the killing process visually, we can also extract additional metrics using the dedicated software application to highlight the killing kinetics. Basically, we can study the killing process and determine different phases, like when the killing process starts and when the maximum killing activity is observed. Besides this, we can move forward and extract other parameters like the killing rate a right number of cancer cells killed by one T cell. Currently, I'm in the process to confirm the encouraging preliminary data already generated using the analyzing softwares. In summary, the Nanolive platform allows me to visualize the killing process and extract killing kinetics information as compared to the classical indirect method used in the lab.
Thank you. So maybe I ask you to expand on that a little bit. So how does actually the use of the live T cell assay help Lightchain Bioscience find your best therapeutic candidates? And whereabouts do you see it fitting in your R&D process?
Firstly, at the initiating phase of a therapy program, the live T cell assay could help us to choose the best time point to analyze the killing, and its time point can be applied in the conventional assay based on indirect method, like the LDH release. Secondly, at lead selection stage, we have a short list of lead candidates to evaluate. The live T cell can help to rank the candidates by analyzing other parameters that we cannot easily extract from conventional methods. The assay format based on a 96 well plate will allow us to test on the same plate different candidates in replicate at different concentration. The throughput of the machine is a key element, and it will be an important feature to evaluate when selecting a technology platform for drug development. Finally, at the candidate comparison stage, the live T cell will help us to reinforce the data package needed to deeply characterize a molecule of interest.
Thank you. So you mentioned in your answer earlier the observation of T cells that then go on to kill multiple target cells. And maybe I can ask you, just out of my own curiosity, we see that referred to in the literature a lot as serial killing, but also I hear the term processivity. So how do you refer to the process of the T cell killing multiple target cells? And how important is that for you when you select your best lead candidates?
Basically, the serial T cell killing is a very important criteria that we need to deeply analyze when we are basically ranking the final candidates, and is a key element to see and to quantify the serial T cell killing in order to probably anticipate in the future the in vivo application to treat real tumors in a live body.
And may I ask just one follow-on question from that? So prior to the use of the live T cell assay from Nanolive, how were you assessing previously the ability of T cells to kill multiple target cells?
Basically, I have to say that currently it's very difficult in order to easily extract this type of criteria based on our, let's say, classical method to quantify the killing. So this is a very advantage to use the Nanolive technology to extract easily the serial T cell killing based on the video analysis.
So I think, Valery, we've taken up lots of your time, and it's been really great to have you here with us today. Thank you so much for your very interesting insights. We wish you, of course, and Lightchain Bioscience continued and great success in producing your therapies. And with that, I would like to say thank you and have a very good day.
Yeah, thank you, Alex. Bye.
Well, with that, we come to the end of the presentations for today. I'd like to thank Valery and our other presenters for being part of this, but really, most importantly, you guys for taking the time to come and see as well. Please remember, you can continue to send us your questions on this meeting, or reach out via email, or contact your local Nanolive rep if you want to find out more. I'm sure that we'll all enjoy all of the questions that you send us, and we'll respond to them as quickly as possible, and that we'll have some even more new exciting material to share with you in the coming future. Thank you very much, and have a lovely day or evening wherever you are in the world.